Welcome back to the deep dive. When you look at the human family tree, specifically going way back into deep time, there's this very prominent silhouette where a hominin called Homo erectus should be.
Right, because we have the physical evidence, right? We have the bones, we have the fossils.
Exactly. We have the physical stuff, but molecularly, it's just been a total blank space.
A complete mystery. Yeah.
But today, thanks to a 400,000-year-old tooth and literally a microscopic drop of acid, we are finally filling in that silhouette.
It's incredible. It's a total paradigm shift.
It really is. We are diving into a groundbreaking new nature study led by paleogeneticist Qiaomei Fu and her team over in China, and it's quite literally redrawing the human family tree.
Yeah, they're doing some mind-blowing stuff.
We're looking at how scientists have finally unlocked the molecular secrets of Homo erectus and, well, discovered that a piece of them might literally be living inside some of you listening right now.
Which is just a wild concept to wrap your head around.
Okay, let's unpack this, starting with the obvious hurdle. So, Homo erectus existed from around 2 million years ago to roughly, 110,000 years ago?
Roughly, yeah. That's the generally accepted timeline.
Right. And we have spectacular skeletons from Georgia. We have those iconic skulls from China. So why has it taken until right now to read their molecular signature?
Well, it fundamentally comes down to the destructive nature of traditional genetic sequencing, clashing with the extreme fragility of ancient biology.
Because DNA just doesn't last that long.
Exactly. DNA is notoriously delicate. I mean, it is highly subject to hydrolysis and oxidation. So under normal environmental conditions, a DNA molecule simply degrades way too fast to survive 400,000 years.
Right.
It literally shears into fragments so microscopic they become mathematically unreadable.
But there's a secondary barrier too, right? Like a logistical one?
Yeah, exactly. Extracting what little genetic material might actually remain, it requires taking a drill to the bone or the tooth and grinding it into a fine powder.
Which museum curators understandably view as an absolute non-starter.
Oh, completely.
I mean, you look at incredibly rare cultural heritage items like the Peking man fossils from the Zhoukoudian site. These are irreplaceable snapshots of human evolution.
Right. You don't just take a power tool to them.
No. You don't drill into them on the 1 in a million chance you might find a surviving fragment of DNA.
Precisely. And that roadblock created a really profound blind spot in paleoanthropology.
Like, we could look at the morphology of these bones all day, but we couldn't actually read their biological code.
Right. And this is where paleoproteomics, which is the study of ancient proteins, completely changes the paradigm. As Enrico Cappellini, a biochemist at the University of Copenhagen, noted in the commentaries we looked at, when the DNA is gone, proteins are our most robust proxy.
Because proteins are the physical manifestation of that genetic blueprint. I like to think of it like the DNA might be a fragile paper schematic that turns to mush after 400,000 years in a damp cave, but the proteins are the actual bricks and mortar built from those schematics.
That's a great way to put it. They weather the millennia far, far better.
But wait, if you still have to get those protein bricks out of a fossilized tooth, aren't you still, requiring physical material? How do you bypass the curator's absolute ban on drilling?
Well, Qiaomei Fu's team utilized this highly ingenious microdestructive sampling approach. It's based on acid etching.
Acid etching, how did that work?
So instead of mechanical extraction like a drill, they wrap the ancient tooth in a tightly sealed waterproof film.
Okay.
And they leave just a tiny exposed window of a few millimeters square. Then they meticulously dab that tiny window with a 5% hydrochloric acid solution.
Oh, wow. So instead of a mechanical drill turning an artifact into dust, it's essentially this highly targeted microscopic chemical coaxing.
Precisely. The acid merely dissolves the absolute most superficial layer of the enamel. It releases the proteins trapped within that specific microscopic sector.
And the tooth is safe?
Yeah. They wash it, they neutralize the acid, and capture the liquid. The tooth remains morphologically pristine. I mean, the only physical footprint of the entire procedure is a barely visible discoloration on a millimeter of the surface.
That is wild. And from that minuscule chemical wash, the team successfully captured, 11 distinct proteins?
11, yeah.
What's incredible to me is how tooth enamel acts as this flawless biological vault.
What's fascinating here is the structural reality of enamel. It is a biomineral matrix. It's primarily composed of hydroxyapatite crystals.
Okay, hydroxyapatite.
Right. So during childhood, as the tooth forms, proteins dictate the growth of these crystals, and then they become locked inside this incredibly dense, non-porous structure.
Oh, so it just seals them in?
It essentially entombs these molecules. It shields them from microbial decay, from groundwater leaching, literally for hundreds of thousands of years.
That is just brilliant. Now, once Qiaomei Fu and her team secured these 11 proteins, they analyzed 6 specific teeth spanning a massive geographic and chronological range in China.
Yes,
right?
A really wide net.
We have Zhoukoudian in the north, up near Beijing. We have Xinjiang in central China and Hexian down south in Anhui province. And all of these date to roughly 400,000 years ago during the Middle Pleistocene.
And the very first demographic data point they pulled from these chemical washes was biological sex.
Wait, really? From just a wash?
Yeah. They targeted a melagenin. It's a protein vital for organizing that enamel crystal structure we just talked about.
Okay, great.
Since the gene for the amyl isoform of this protein is located on the Y chromosome, tracking its presence against the more ubiquitous AMYLX variant gives you a highly reliable sex typing mechanism.
Right, and the researchers used a computational method called Prout-Sex-and-Per to calculate that ratio. But I actually want to pause on the method itself for a second. Why do we need a complex algorithmic pipeline for this? If you have the chemical wash, aren't you just sort of looking under a proverbial microscope to see if the ABLE protein is floating around in there?
Well, if the proteins were perfectly intact chains, yes. But remember, these biominerals have been sitting in the earth for 400 millennia.
Oh, right. So they're degraded.
Very degraded. The proteins have undergone diagenesis, meaning they've broken down into chaotic, jumbled peptides. It's basically chemical noise at this point.
Okay.
You aren't looking at a clear protein sequence. You're looking at fragments. So Prout-Sex-and-Per, alongside other mass spectrometry analysis pipelines like MassQuant, Peaks Online, and pFind, they act as sophisticated pattern recognition engines.
I see.
They statistically reconstruct the amylite to AMYLX ratio from that degraded chemical noise. And using this, they found 5 of the teeth were male, and 1, an adult from the Xinjiang site, was female.
Okay, I have to push back a little on the reliability of identifying anything from 400,000-year-old chemical noise.
Fair enough.
Given how fragmented these peptides are, couldn't a microscopic fleck of modern contamination completely skew the algorithms? Like, what if a lab technician casually talking over an open sample introduces modern amyloid proteins? Couldn't that create a false male positive?
That intense skepticism is exactly why paleoproteomics is so strict. It's a foundational requirement. They don't just rely on the presence of the amino acids, they actually analyze their chemical degradation.
The damage is the proof.
Exactly.
Yeah.
Specifically, they look for a natural phenomenon called deamidation.
The structural breakdown of the molecules over deep time.
Right. Over thousands of years, specific amino acids like glutamine and asparagine spontaneously shed ammonia molecules when exposed to trace environmental moisture.
Okay.
It is a slow, relentless, and completely unfakeable thermodynamic process, a modern protein introduced by a sneezing lab tech. It would appear structurally pristine.
It wouldn't have any of that damage.
Zero deamidation. But the proteins extracted from these 6 ancient teeth exhibited massive deamidation levels.
So the chemical damage itself is the unassailable proof of authenticity. You were looking at a genuine artifact of the Middle Pleistocene precisely because it is falling apart.
Authentic and untainted. And once that baseline of authenticity was locked in, the team moved beyond basic demography to look at specific amino acid variants within these proteins.
The subtle genetic typos that act as inherited molecular fingerprints.
Exactly.
And they found 2 really profound mutations. Let's look at the first one, which is located in a protein called ameloblaftin. It's a variant called AMBN A253G.
Right.
They found this specific mutation across all 6 teeth, from the freezing caves of Zhoukoudian all the way to the southern climates of Hexian.
And the significance of AMBN A253G is its absolute exclusivity. It is a molecular signature that has never been recorded in any other primate or hominin lineage.
Never.
Never. Not in modern humans, not in Neanderthals, and critically, not even in Homo erectus specimens from Georgia or the Homo antecessor fossils in Spain. It belongs entirely to this specific group.
Here's where it gets really interesting. Because identifying this unique signature definitively solves a raging, decades-old archaeological debate regarding the Hexian fossils.
It really cuts straight through all the morphological confusion.
Right, because for years, paleoanthropologists noted that the skulls from the southern Hexian site just looked different from the famous Peking Man skulls in the north.
The bone structure was visibly distinct, yeah.
And that led many to hypothesize that the Hexian individuals might be a completely different lineage, like maybe an offshoot of Indonesian Homo erectus, or even an early Denisovan population.
But morphology can be incredibly deceiving. Bone shapes shift based on diet, climate adaptation, or just isolated local traits.
Right, so this unique ameloblastin mutation acts like a highly specific family crest.
That's a perfect analogy.
Because all 6 teeth share this exact unprecedented variant, it proves that despite their physical differences and massive geographic separation, they belong to the exact same evolutionary population.
It provides the very first irrefutable molecular marker for East Asian Homo erectus. As Yale geneticist Denisovans pointed out in his commentary on the study, having this unique variant creates a sort of tracking beacon.
Oh, that's cool.
Right. Paleoanthropologists no longer have to guess relationships based on the curve of a brow ridge. We can use this protein signature to trace the exact migration routes and boundaries of this specific lineage across the Eurasian continent.
That makes total sense. That first mutation draws a boundary, proving who this distinct East Asian group was. But the second mutation Qiaomei Fu's team isolated does something much more expansive.
Yes, it does.
It connects this 400,000-year-old population to the wider human network. The variant is called AMBN M273V.
And finding AMBN M273V in Homo erectus is a massive plot twist. I mean, until this Nature paper was published, paleogeneticists considered that specific variant to be the definitive molecular signature of the Denisovans.
Which changes our entire understanding of Denisovan biology. I mean, we know Denisovans are a deeply elusive group. They're known almost entirely from fragmented DNA, extracted from just a handful of teeth and bone shards in Siberia and Tibet.
Right.
So if we thought this tooth mutation was uniquely theirs, finding it in fossils that predate them by hundreds of thousands of years means they didn't invent it.
They inherited it.
Wow. And since we already know Denisovans subsequently interacted and interbred with migrating Homo sapiens, this creates a direct molecular bridge.
Exactly.
Denisovans inherited this tooth variant from Homo erectus and then passed it along to modern humans. The study actually notes this exact variant is still present in living populations today.
Yeah, particularly in Southeast Asia and Oceania.
Right. It appears at a 21% frequency in the Philippines alone.
If we connect this to the bigger picture, this protein variant provides the first hard physical evidence for a theory geneticists call superarchaic introgression.
Superarchaic introgression. Okay, break that down for us.
So when scientists first sequenced the Denisovan genome, they found an anomaly. Roughly 8% of their DNA appeared to originate from a ghost lineage, a superarchaic hominin group that had diverged from our shared ancestors over 1 million years prior. We could see the statistical shadow of this ghost in the code, but we had no actual physical evidence to identify who they were.
And thanks to this tiny drop of acid on a fossil tooth, the ghost finally has a name.
It does.
Homo erectus is the superarchaic population that left their mark on the Denisovans. It vividly illustrates that human evolution was never just a neat sequence of one species replacing another.
Not at all.
It was a highly porous, tangled bramble of different hominin groups, overlapping, migrating, and mingling across deep time.
It's beautifully complicated.
But wait, I want to pause and pressure test the assumption here for a second.
Sure, go ahead.
Just because 2 ancient populations possess the exact same amino acid variant in their tooth enamel, does that automatically guarantee a shared evolutionary legacy? Like, why couldn't this be an instance of convergent evolution?
How do you mean?
Well, it's like 2 isolated software engineering teams independently writing the exact same highly complex algorithmic patch for a system vulnerability without ever actually communicating. Technically possible, right? Couldn't Homo erectus and Denisovans have just separately developed the same beneficial tooth mutation?
You are tapping into the core scientific debate surrounding this discovery right now. And the software engineering metaphor is actually highly appropriate here. Denisovans, the Yale geneticist we mentioned earlier, he champions a necessary skepticism on this.
Right. He says we shouldn't jump the gun.
Exactly. He argues that while the paleoproteomic data itself is flawless, jumping to the conclusion of ancient interbreeding remains a hypothesis until we have a complete Homo erectus genome.
Because without mapping the entire code base, we cannot definitively rule out independent convergent evolution.
Right. It's the scientifically rigorous stance. You don't declare a definitive genetic crossover without seeing the full genome.
Right.
But on the other side of the debate, University of York geochemist Kirsty Penkman and biochemist Enrico Cappellini offer a very compelling biochemical counterargument.
Based on the constraints of the proteins, right?
Exactly, based on the extreme physical constraints of these specific proteins. Penkman's argument hinges on the fact that ameloblastin isn't just a random biological trait. It is a foundational biomineral.
Okay, so it has to follow strict physical rules.
Very strict. These proteins have an incredibly rigid structural mandate to construct the crystalline lattice of tooth enamel. If a mutation alters the shape of the protein too drastically, it fails to interlock with the hydroxyapatite crystals.
Oh, I see.
And if that happens, the enamel becomes brittle, the tooth fails, and in a Pleistocene environment, a hominin with failing teeth is facing a severe evolutionary dead end.
Wow. Okay, so the biochemical constraints mean there are very, very few ways for this protein to mutate without becoming lethal.
Precisely. Because the protein is under such strict functional limitation, the statistical probability of two separate hominin lineages rolling the exact same genetic dice to arrive at the precise AMBN M273V mutation independently, it's astronomically low.
It's just too much of a coincidence.
Way too much. When you combine that sheer biochemical improbability with the geographic and chronological realities, the fact that Homo erectus and Denisovans were cohabitating in the exact same East Asian environments during the same geological epochs, a shared evolutionary legacy, isn't just a theory anymore.
It becomes the most logically sound conclusion.
Right. The structural constraints of the protein heavily point to interbreeding rather than independent parallel evolution.
So what does this all mean? We started this deep dive staring at a blank silhouette in our evolutionary history. And thanks to an ingenious waterproof film, a localized acid wash, and the astonishing durability of a biomineral matrix, that silhouette is finally stepping out of the shadows.
It really is.
The journey from a Middle Pleistocene cave in Zhoukoudian to the modern populations of Melanesia and the Philippines proves that our history is beautifully, wonderfully messy.
So messy.
I mean, if you have ancestry from Southeast Asia or Oceania, you might literally carry a microscopic echo of Homo erectus in your biology today.
And for me, the most exhilarating aspect of this breakthrough is the realization of just how much uncharted territory remains. I mean, as Penkman highlighted in her commentary, paleoproteomics is just crossing the starting line.
Really? How much have we actually looked at?
Thus far, fewer than 20 ancient hominin specimens have been analyzed utilizing the specific micro-destructive methodology.
Wait, barely 24 individuals? Out of millions of years of hominin history sitting in museum archives worldwide.
Barely 24, which means we are standing at the edge of an entirely new frontier of molecular archaeology. Every previously off-limits fossil is now a potential data point.
That is just staggering to think about.
This raises an important question. If this single isolated protein variant survived this staggering genetic journey from East Asian Homo erectus, integrating into the Denisovans, and ultimately weaving itself into modern human populations, what other molecular ghosts are currently hiding in our collective genome?
That is a great question.
We know Homo erectus was an incredibly successful, highly mobile species. We have their pristine fossils waiting in the archives of Africa. We have specimens resting in Europe.
Entire lineages we haven't even tested yet.
Right. What completely unknown evolutionary bridges might we discover when Qiaomei Fu's non-destructive technique is applied to those populations?
The possibilities are endless.
The ghosts of our ancestors aren't merely trapped in the sediment of ancient caves waiting to be cataloged. They are woven directly into our own biology, just waiting for the right chemical wash to finally speak.